Hi everyone, this is Scott Reeves and this is FSN 329. Today we're going to go through B12. I want to start with B12 reading you a case study. Now we've already talked about B12 some in when we talked about folate metabolism, but so we'll see some more details on that with overlaps in folate. But one of the other issues, some of the other issues with B12 deficiency are indicated in this case study. So I found this online a while back, and so it starts off like this. This person was 30 years of age at the time that she wrote this. She says, "I'm happy to help in any way I can. I would hate for anyone to go through what I did. It started when I felt like I was having shin splints in my legs, and I didn't think much about it at first. Then my legs started getting weak and they would shake when I went up and down stairs and things. The aching increased until it was constant and I had sharp pains in them as well. I kept saying it felt like nerve pains. They also started to feel bruised. The pain got so bad I was taking Tylenol and Advil every 2 to 3 hours just to get through a day. My walking got really bad. My legs just wouldn't work right and I had to convince myself they would walk. On March 4th, I went to the doctor and the nurse practitioner said my legs were tired and prescribed anti-inflammatory medicine. Within 2 weeks, my arms became involved as this whole process has been going on for several months now. They hurt, felt bruised, and I had no finger coordination. I had no strength. I could not hold a cup or open jars. I spent most of my time lying around praying for a day without pain. I had almost lost all function in my arms and legs, and the pain was indescribable. On March 18th, I returned to the doctor and saw my doctor, capital letters. She suspected MS, extensive, so she suspected multiple sclerosis. Extensive testing was done to include blood work and the serum B12 level and MRI of the brain, which ruled out MS. On March 18th, the serum B12 level was 246 picograms per mil, which was just below the normal range. The folate, a little bit low in folate. Hematocrit was normal. And mean corpuscular volume, which is red blood cell size, was actually in the normal range as well. On March 22nd, I started weekly B12 injections, and on May 1st, the injections were decreased to every other week. I noticed with the injections, it took about 6 months for my nerves to heal. An EMG showed no permanent damage, for which I was told I was very lucky. I do still have some fine motor skill problems. My hands cramp some when I use them a lot, and my legs will weaken if my B12 goes too low. My energy level is still not at all what it should be, but is much better now than it was a year ago. I am not a vegetarian. This illness just came out of nowhere, and it was really awful. Signed, KF from Missouri. So this is what we talked about a little bit before. We said that one of the symptoms of B12 deficiency has to do with nerve implications or neurological disorders. And as this individual mentioned, sometimes those disorders along the nervous system, they'll tend to start on the periphery and then they'll work inward, but they can be irreversible. So it's important that B12 deficiency is treated immediately and that it's diagnosed properly. So we're going to talk some more details about that, but one of the things that you should take from this was, right, she's 30 years of age, so age isn't really an issue, and normal blood work as far as red blood cells. So she didn't have megaloblastic anemia, and yet she had nerve problems. So sometimes in B12 deficiency, you don't have to have megaloblastic anemia. So sometimes the blood symptoms are not there, but the neurological complications are occurring. So it's important that they're treated. Okay, so let's go through this. So a little bit of history on vitamin B12. Now, obviously, this is a water-soluble vitamin, but some of the characteristics are a little bit different than the water-soluble vitamins that we talked about before. Now, megaloblastic anemia we've talked about. It was first described in the 19th century, so this one goes back a while, and it was called pernicious anemia because of its invariably fatal outcome. In the 1920s, Minot and Murphy described the first effective treatment, and their treatment was to have patients with pernicious anemia eat 1 pound of raw liver a day. 1 pound of raw liver a day. And for this treatment plan, these two individuals received a Nobel Prize. Okay, so what does that tell us? That if you want to win a Nobel Prize, years ago when you were in nutrition, have your patients eat raw liver. And that could help, could get you a Nobel Prize. The other thing that we should take from that is that's one of the main storage sites of B12 in the body is the liver, okay, for lots of different animals, including us. Now, it later became apparent that a stomach factor was required, and that stomach factor was required to process something coming from the diet. Because the stomach factor was made within our body, they called that intrinsic factor. And it was made to process something in the diet that they termed extrinsic factor. Okay, originally it was thought that folate was the extrinsic factor. And then they eventually realized it was actually B12. Now what's the intrinsic factor? Well, that's what we now know as intrinsic factor. It's actually a protein that's made in parietal cells of the stomach, and it's a small, relatively small glycoprotein that's required for B12. So individuals with pernicious anemia were not synthesizing or producing adequate amounts of intrinsic factor, and that's why they couldn't process or absorb the extrinsic factor from the diet. We'll talk more about pernicious anemia coming up. So here's B12, and it's one of the more complex vitamin structures. And this diagram doesn't really show it justice because it actually has a really unique three-dimensional type structure to it. If we look up at the top here, we can see this kind of ring, this elaborate ring structure here. And in the center of this ring is a cobalt atom. Cobalt. And in this case, attached to that is a cyanurin. And this can be replaced, but it's always a cobalt. Okay? This planar structure actually kind of flattens out and is juxtaposed to this lower ring structure, so it's kind of two flat planar components to this. So it kind of has almost like two stories or layers to it. The structure of this causes it to have a really bright, beautiful red color. Actually, if you see vitamin B12 in a jar isolated, it's actually a beautiful red color. So sometimes you'll hear people, classic nutrition papers, they'll say it's the most beautiful vitamin or something along those lines. You look down here, it also has an amine group. There's a few amine groups. So originally they started calling this cobalamine because they knew it had cobalt and it has an amine. And this ring structure overall up here is called, this is a corin ring structure, C-O-R-R-I-N. And so that's one of the unique parts of this. So now what's interesting about B12 is that cobalt is always there. The cobalt has a few different valence states, but that cobalt, so here this kind of rectangle structure here represents that corin ring structure, the cobalt in the middle. Now that cobalt can attach to a variety of other chemical groups. For example, it can bind to hydroxyl groups. And here is, don't worry about this part, this is just the bottom part of it. They're trying to simplify that. That actually stands for 5,6-dimethyl benzoimidazole. So don't worry about that. Now, in this case, that's called hydroxycobalamin. That cobalt can also be attached to an adenine. Here's the ribose, and here's an adenine structure. Sorry, it goes through ribose to this. If this is attached to the cobalt of the B12, it's called 5' deoxyadenosylcobalamin. So it's always cobalamin, and here is the other chemical group that's attached, 5' deoxyadenosylcobalamin. Okay, for short, we can call this adenosylcobalamin. Okay, here's our ribose, adenine, here's adenylyl, and the cobalamin. That's one of the two active forms. If it's a methyl group, we call this methylcobalamin. If it's a methyl group, that is another active form. So we want to remember that the two active forms of B12 are 5' deoxyadenosylcobalamin and methylcobalamin. Okay, what that means active, that means they function as coenzymes in reactions. If there's nothing attached, it's just called cobalamin. If there's a cyanin group attached, it's called cyanocobalamin. Okay, that's a common form. So cyanocobalamin is the most common form found in supplements and enrichment and fortification scenarios. It's cyanocobalamin. Now our body can take in pretty much any of these forms and cyanocobalamin and convert them to the active forms. Okay, so we'll see a few different forms in the diet that our body can actually convert to these two active forms, so that's not a problem. Okay, let's look at the other one. Next slide. Plants do not make B12. Animals do not make B12. Only microorganisms can make B12. Animals obtain their B12 from microorganisms, and then we consume the animal products. I think we've all probably heard this, that the only sources of B12 are animal products, and tend not to be in plant foods, plant-based foods. So we've probably all heard, vegetarians need to be careful because they have to have good B12 diets, and in general, vegetarian levels of B12 in the blood do tend to be lower than those individuals that are not vegetarians. But there are quite a few different food sources where vegetarians can pick up B12, those food sources that have been fortified, or of course they can take vitamin B12 supplements, which I would say is probably recommended. That's a solid recommendation. But a lot of people think, well, animals make B12 because they're good food sources, but they're actually not. They actually don't make B12. Okay, so we're going to simplify this, and we're going to say the best sources include animal products, which would include quite a few of these. So we're just going to say animal products and fortified cereals. That tends to be the most common sources in the U.S. Remember, B12 is not part of the enrichment program, so these are fortified products, especially fortified cereals. So strict vegetarian diets contain very little B12, and many vegetarians have low plasma B12 levels. However, here's an important point. Most cases of B12 deficiency are due to compromised absorption rather than inadequate levels in the diet. So most cases of B12 deficiency are due to compromised absorption of B12. Okay, here's some examples, sources of B12 in foods, and these are all standardized by, this is the amount of vitamin B12 in micrograms per 100 grams of each of these food sources, so we can kind of see relative amounts. If we look up here, for example, we have meats like beef, we have 1.94 versus 3.64, and to get an idea of whether or not this is a significant amount, I'll just tell you that the RDA is 2 micrograms, is around 2 micrograms. Okay? So if we look at, for example, beef brain, we all love that, 7.8 micrograms. Beef kidney has 38. Look at beef liver, 69 to 122 micrograms per 100 grams. Remember we said that their solution for pernicious anemia was eating 1 pound of raw liver. Well, look at all the B12 in there relative to other food sources. Even chicken liver is high compared to the other sources. If we just look at the regular ham or pork or turkey products a lot of people consume, you look at that, seems like low amounts, but relative to the RDA, those are good sources. And down here at dairy products, there is B12 in these, including milk and cheeses, a little bit in yogurt. And then over here, fish tends to be a relatively good source. Now, some of the mussels, clams, and oysters are really good sources. Lobster, shrimp, eggs are good sources. If we look at this, you can see that most of the B12 is actually in the egg yolk, and that's a significant amount. That's quite a bit there in that egg yolk. And then we see vegetables, grains, fruits. None of those contain B12, the last item here. Okay. Now, B12 absorption. So I want to go through this and give you the steps. And so you should be able to give me back these details on this. And the reason we're going to go into a lot of detail on this is it's a relatively complicated process for B12 digestion or absorption, and there's one or a couple things that can go wrong, and that's why B12 deficiency is actually relatively common. Okay, so we have this diagram. Give me 1 second. Okay, so let's continue on here. And give me 1 second. Okay, so the steps in B12 absorption are shown here. So in step number 1, the B12 is released from food proteins by hydrochloric acid and pepsin in the stomach. So most B12 in food is going to be bound to protein. So as it goes into the stomach, it needs to be separated from those proteins. Now obviously supplemental B12 is different because it's not bound to proteins. But in the case of food protein, it is. Okay? So the B12 is released from proteins in the stomach. And in the next step, R proteins, and that's R, is the letter R, R proteins made in the stomach bind to B12 as it's separated from the food proteins. In the stomach, intrinsic factor is released by parietal cells. Okay? The B12R protein complex moves through the stomach into the small intestine. Intrinsic factor also enters the small intestine. The R proteins in the small intestine are digested. That releases B12. The B12 then binds intrinsic factor in the small intestine. The B12 intrinsic factor complex moves through the small intestine until the ileum. In the ileum, the B12 intrinsic factor complex is recognized by a receptor called cubulin, and that's spelled C-U-B-U-L-I-N, and that's what's represented right here where it says ileal B12 intrinsic factor receptor. That's cubulin. The B12 intrinsic factor complex is absorbed by receptor-mediated endocytosis. So in other words, B12 intrinsic factor and cubulin enter the cell in the ileum. That complex is contained in a vesicle. A vesicle, V-E-S-I-C-L-E. The vesicles enter the intestinal cells and fuse with a lysosome. Lysosomal enzymes degrade intrinsic factor, and B12 is released into the cytosol of the cell. Some B12 can be used by the cell, and some B12 is released through the basolateral membrane into the portal vein where it binds a protein called transcobalamin 2, and that's shown on this slide, the transcobalamins. There's 3 of them, and as B12 leaves the enterocyte or the cell in the ileum, it's bound to transcobalamin 2. Transcobalamin 2 transports B12 to the liver, where it's taken up by receptor-mediated endocytosis. Okay, we'll talk about transport in 1 second. So overall, a couple things here. The overall absorption of B12 is about 50%. So about 50% of dietary B12 is normally absorbed. Okay, so one of the questions is, why do you have R proteins and intrinsic factor? Well, B12 tends to be needed by microorganisms in our gut. Right? And so it's thought that maybe the R proteins keep B12 from being used by bacteria in the stomach. And then when it goes into the small intestine, it's thought that intrinsic factor might keep microorganisms from bacteria basically, being able to use B12 as it goes through the small intestine. Okay, we'll talk about more of this later, but that's maybe one of the reasons we have R protein and B12. Now, why does the R protein release B12 when it gets in the small intestine? Now, R protein obviously can bind B12 at a low acidic pH. And that's what's happening in the stomach. Now, intrinsic factor is not binding in the stomach. It could be because at that low pH, intrinsic factor can bind B12. But now in the small intestine, when the pH elevates to approximately normal pH, that R protein is going to release B12. It's going to be degraded by digestive enzymes. Now the intrinsic factor can actually bind the B12. So it could be like, I read a couple places where it could be pH dependent, and then also once those digestive enzymes in the small intestine degrade R protein, then intrinsic factor is now able to bind. But again, there's a few things that have to happen in order for B12 to be absorbed into our ileal cells and actually be used by the body. And we'll see that in pernicious anemia, intrinsic factor synthesis is actually compromised. Okay? So now, transport and storage. Overall, I'm looking at third bullet point, but overall about 80% of the B12 in the blood is bound to transcobalamin 1. And an exception would be as it's going through the portal vein to the liver, it's transcobalamin 2. But the rest of it is usually transcobalamin 1. Now, here's where B12 is unique among the water-soluble vitamins. About 2 to 4 milligrams of the vitamin is stored in the body and is stored mainly in the liver. Now, if we stop consuming B12 today, it would take a few years for us to actually show signs and symptoms of a B12 deficiency. And that's one of the reasons it's so unique among the water-soluble vitamins. It takes years of inadequate B12 levels before signs of a deficiency or complications of a deficiency actually manifest. So our body does a really incredible job of efficiently retaining B12 to help protect against a deficiency. However, it does happen. Okay? Because if you do have compromised absorption for a few years, and something in your physiology becomes abnormal or changes, then eventually it can lead to a B12 deficiency. One of the issues here is B12 is actually, there's some B12 in the bile, and that B12 actually leaves the liver, goes into the bile, goes through the gut, but most of the B12 is reabsorbed. So even though it's there, it actually is efficiently reabsorbed. Now, we've talked a lot about B12. We've said it's a cofactor or coenzyme for reactions in the body, and overall the symptoms of B12 deficiency are can be pretty harmful. But with all that said, there's only two enzymes in the body that actually require B12, and one is methionine synthase. We've talked about that one a lot already. We'll talk more about it. The other one is an enzyme called methylmalonyl-CoA mutase, and we'll talk about that one as well. Those are the only two enzymes that require it. So let's go into a little bit more detail, and if you want to title this slide, you could basically probably put methionine synthase. And we've seen this before. This is slightly different detail though. So here's THF, bottom left. THF is shown here, so remember you have this same slide in your folate lecture, but also remember on that folate metabolic pathways chart, THF's kind of right in the center of the diagram. And then it goes from THF to 5,10-methylene THF. And we looked at this, it's a serine hydroxymethyltransferase. You don't have to remember that enzyme, but remember a methyl group is now added to THF, and the methyl group comes from serine. So now we end up with glycine, and we end up with 5,10-methylene THF. That molecule is converted to 5-methyl THF by methylene tetrahydrofolate reductase. We talked about that enzyme. So a point here is bidirectional reaction down here, unidirectional reaction going from 5,10 to 5-methyl THF. And remember we talked about this reaction. We said during this reaction, it's unidirectional, generates 5-methyl THF. If this enzyme activity is compromised, that's one thing that can lead to reduced levels of 5-methyl THF. And we also said if B12 is deficient, it can lead to this molecule accumulating. We called that the 5-methyl THF trap. And the reason is here. 5-methyl THF is used by methionine synthase. Methionine synthase uses B12 as its coenzyme. Okay, so here's B12 in its cobalamin form. And remember, cobalamin can actually become methylcobalamin by picking up a methyl group. And the methyl group comes from 5-methyl THF. So now we have methylcobalamin, one of the active forms of B12. This enzyme picks up the methyl group here, transfers it to homocysteine, and now homocysteine becomes a methionine. Now the enzyme now has cobalamin, so it could pick up another 5-methyl THF if it's available. But remember, B12 isn't present or isn't in adequate amounts, then the activity of methionine synthase is compromised because it needs cobalamin, it needs B12 to take this methyl group from THF, from 5-methyl THF. So no B12, none of this, and, no reaction with 5-methyl THF, and no conversion of homocysteine to methionine. One of the things we talked about before was we said homocysteine is actually harmful. And I gave you 3 reasons why. But here's more detail to this particular aspect. Once we have methionine, methionine can be converted to a molecule known as S-adenosylmethionine. So as its name implies, an adenosine group has been added to methionine. And the enzyme that does that is here. You don't have to remember that enzyme name. This is methionine adenosyl transferase. Takes ATP, takes the adenosine group off of it, and adds it to methionine. Okay? So what's happened is we made methionine, and now methionine is being converted to homocysteine. It goes back to homocysteine in these steps. So you want to remember it goes from methionine to S-adenosylmethionine. This adenosine group has a methyl group that can be added to other molecules. So the methyl group from methionine is added to acceptors of methyl groups. And there's quite a few different molecules that accept that methyl group. Okay, so see where this says CH3 methyl acceptor? I want you to put next to that, for example, myelin basic protein. We've all heard of the myelin sheath. All right, we'll talk more about this, but the myelin basic protein, that's MYELIN, basic protein is one of the proteins that requires methyl groups from S-adenosylmethionine. So this molecule is required to make myelin basic protein. All of this requires B12. So one of the theories behind the cause of neurological dysfunction in B12 deficiency has to do with this step right here. And we'll talk about that. Now, once we convert that, now we lose the methyl group from methionine. Now we have S-adenosyl homocysteine, which is homocysteine with this adenosine still attached. And then the adenosine is taken off, we end up with homocysteine. And you can get rid of this last part here. So you want to be able to basically draw this diagram, and on this side of the diagram, just put methionine to S-adenosyl methionine, and then put CH3 acceptors such as myelin basic protein, and then that converts to S-adenosyl homocysteine and then homocysteine. That's it. You don't have to put these details with adenosine here, and you don't have to include the bottom part. Okay? Pretty interesting the way all that works. Now let's talk a little bit more about deficiency of B12. Remember, we just said that B12 deficiency leads to megaloblastic anemia, and basically it's because that 5-methyl THF trap causes alterations in folate metabolism, because folate tends to accumulate as 5-methyl folate. And when that happens, DNA synthesis is compromised. So if we look kind of in the middle here, these are normal red blood cells. This is used in a stain called Wright stain. We're actually going to do this in a couple weeks in lab. That's what normal red blood cells tend to look like. Over here are megaloblastic red blood cells. They're large, they're pale, they're dysfunctional. They are not transporting oxygen adequately. They tend to have abnormal structure. And they just don't function. This is not an abnormal cell. This is a white blood cell. So all these kind of pale looking cells here are evidence of megaloblastic anemia. Okay? So what this causes, megaloblastic anemia actually causes, decreased energy production in the body. So one of the classic signs of deficiency is the person is tired or easily fatigued or easily becomes tired because the cells aren't delivering oxygen the way they should. So as far as hematology, it's the same as folate deficiency. Either one of them will cause this. Okay, so it's important that we differentiate between whether or not a person is B12 or folate deficient. So if anemia is present, must determine it's due to B12 or folate. Plasma homocysteine levels can be increased by both folate or B12 deficiency. So you can't just look at homocysteine levels. So one way to do it would be looking at serum methylmalonic acid. That would only be elevated by B12 deficiency. So measuring serum methylmalonic acid is considered to be an important indicator of B12 status. So how would you confirm B12 deficiency? Well, you can measure serum methylmalonic acid, then give the patient or person B12, and then do repeated analysis of serum methylmalonic acid and see if it's actually been changed by the B12 addition. And if it has, then that's an indicator probably B12 deficient. Now, you can also look at B12 in the blood or serum and measure that directly, and that can be used as an indicator, and that's typically what people do or clinicians do. But a lot of people think that measuring this serum methylmalonic acid is probably a better indicator or more sensitive. We'll come back to that. Actually, let's look at that right now. one of the things that we've said with actually, let's go to this. This is B12 and neurological function. We looked at some of the reason. We said, well, myelin basic protein might be the mechanism that through which B12 deficiency causes neurological dysfunction. So in this case, we have neurons with a myelin sheath. So not all neurons have myelin sheaths, but some of them do. And the myelin sheath is a combination of proteins and lipids that are around a part of the neuron, and the myelin sheath can actually help the efficiency of signal transduction in the membranes of these neurons. Okay, so it's important that it's there and functioning properly in some neurons. If there's damaged myelin sheath, it might look something like this, and that can compromise the neurological function of that tissue. Here's another way of looking at it. This is actually one of the better ones I could find, believe it or not. So these are cross sections of neurons, and this kind of layer out here is supposed to represent myelin, and it's the sheath that's incorporating that. So myelin basic protein would be a protein that's actually part of this myelin sheath, this green area. And when its synthesis is compromised in B12 deficiency, that might be why B12 deficiency leads to neurological function, compromised neurological function. Okay, so that has to do with methionine synthase. The second enzyme that is B12 dependent on our body is one that is called methylmalonyl-CoA mutase, and it's shown down here. The reaction that it performs is converting methylmalonyl-CoA to succinyl-CoA. Right here. Now, this enzyme requires adenosylcobalamin. Methionine synthase requires methylcobalamin. Methylmalonyl-CoA mutase requires the other active form of B12, and that's adenosylcobalamin. Okay? So how do we get here, though? If we look up at the top of this diagram, we see propionyl-CoA. You might not have ever heard of that, but propionyl-CoA is very similar to acetyl-CoA, except it has 3 carbons. During the oxidation of carbon skeletons of methionine, threonine, isoleucine, and or beta oxidation of fatty acids with odd-numbered carbon chains, you get propionyl-CoA. So you could say, during the oxidation of some amino acids and fatty acids, propionyl-CoA is made. Okay? Now, that's converted to D-methylmalonyl-CoA, which is converted to L-methylmalonyl-CoA. So I'm fine if you just say it goes from propionyl-CoA to methylmalonyl-CoA and then succinyl-CoA. And this indicates the B12 dependent enzyme known as methylmalonyl-CoA mutase. So here's what happens. If a person is B12 deficient, the activity of this enzyme can be compromised. That causes methylmalonyl-CoA to accumulate. That converts to methylmalonic acid. Basically, the coenzyme A comes off to become methylmalonic or methylmalonyl acid. Because it doesn't have CoA, it can leave the cell, so it'll end up in the urine, it'll end up in the blood. So it can measure either one. So when that starts accumulating, that's an indicator of probably B12 deficiency. So that's why it can be used for that. Okay, I think that's all I have to say there. Now, the RDA. Remember, so the DRI for vitamin B12 is an RDA. And the recs are based on estimates of vitamin intake and turnover, and on the amounts of the vitamin needed for the maintenance of normal serum indices of the vitamin and hematological status. So when they determined the RDA, they looked at this type of data. So they looked at vitamin intake and turnover, and B12 turnover is very, very slow. We said that. Just a little bit can stay around in our body for long periods of time. But how much do we need? Well, you're looking at normal serum indicators of the vitamin, So normal amounts of the vitamin in the blood and red blood cell formation. So the RDA using that data was established at 2.4 micrograms per day. That's a really small amount. That's all you need, 2.4 micrograms. So far we've been talking about milligrams in recommended amounts. Now we're at micrograms. For RDA for pregnancy and lactation is 2.6 and 2.8 micrograms, so it goes up a little bit. And the requirement, in other words, the EAR for adults is 2 micrograms per day. Then we add 0.4 microgram to get to the RDA. Now, here's an important point. It is suggested that persons 51 plus years old consume foods fortified with B12 or take B12 supplements because 10 to 30% of older individuals have changes in their GI tract that limit their ability to absorb food-bound forms of the vitamin. That's a pretty significant number, and some people think that that's conservative. As we get older and things in our gut change, like the amount of hydrochloric acid production, that would limit the ability to separate B12 from proteins in the stomach. There are also conditions where the inside of the stomach lining, or the lining on the inside of the stomach, becomes inflamed and dysfunctional. So we have less intrinsic factor that's made. And that can cause inadequate B12 absorption as well. Now, if you take it from supplements, you don't have to separate it from the protein because in the supplement it's not bound to a protein, so absorption becomes easier. So that is sometimes used as a way of ensuring that as we get older, some of the changes in our gut don't cause a B12 deficiency. Now remember, as we get older, some of the symptoms that I read you from that case study, a lot of people assume, oh, that's kind of a normal part of aging. You know, I can't use my hands as well as I used to. I get, you know, I get some pain in some of my, you know, joints or extremities, or, but it can be a B12 deficiency. So it's important that we realize that, or that people are made aware of that they should get their B12 checked, especially as we get older. So signs and symptoms of deficiency include fatigue, shortness of breath, palpitations, insomnia, tingling and numbness in extremities, abnormal gait, loss of concentration, memory loss, disorientation. Again, sometimes we attribute that to getting older, swelling of myelinated fibers, and possibly dementia. So there's quite a range of these that can be caused by a B12 deficiency. Neurological problems occur in about 75 to 90% of deficient individuals. That's a very, very high percentage. Okay, so most of the time there's going to be neurological complications. A lot of times it happens with no anemia. Sometimes it happens with anemia, sometimes it doesn't. Okay. Degeneration of spinal cord and peripheral nerves tend to occur. Again, tends to start on the outer extremities and then work its way in. But it's important that it's treated right away. Can be severe and irreversible. Hyperhomocysteemia can also result. Remember we talked about that, how all that works. So again, most cases are due to inadequate absorption. So if we look at this, here's an example, potential causes of B12 deficiency. Inadequate intake, such as situations where a person's consuming a plant-derived unsupplemented diet, can lead to inadequate intake. Okay, there's obviously other situations, but that's kind of one of the more common ones. Now, here are some things that can cause impaired absorption. Lack of intrinsic factor. Pancreatic insufficiency. In other words, if the pancreas is not functioning properly, it can compromise protein digestion. And so the B12 remains attached to the protein. It's not absorbed. That's one of the reasons that's so important with B12. Intestinal parasitism. Not as common in the U.S. as it is in developing countries. There's reason to believe that B12 deficiency is a lot more common in developing countries. And but there are situations here. Kind of a classic B12 story is, boy, years ago, early 1900s, it was more of a problem, especially in certain cultures. Like, for example, it was found in numerous Jewish women, and they finally figured out that what was happening is they would get together at these events or family get-togethers, and they would make traditional Jewish food, including gefilte fish. And when they were doing this, they were using the type of fish that they would start with raw fish, and it was a slow cooking process. They would add seasonings and different components to the dish throughout the day, but as they were going along, they were tasting it. Some of the fish that they were using were notorious for having parasites and the larvae of parasites. So they would consume those, and then those larvae developed into full tapeworms. And they say they could have, some of them had tapeworms that were 30 or 40 feet long in their gut. And as the B12 went through the gut, the tapeworm would take all the B12, so none of it was being absorbed. And so they found that was something that was causing a B12 deficiency. Not great. Another thing is drug treatment. There are lots of individuals that take different types of drugs, for example, to decrease hydrochloric acid production in the stomach, and that helps them with conditions such as GERD and a few other conditions. That decrease in hydrochloric acid can compromise B12 absorption. Okay, if we look down at the bottom, come back to this lack of intrinsic factor. For example, certain types of stomach operations can affect the amount of intrinsic factor. Right? So if you have less surface area of the stomach because part of the stomach has been removed from the absorptive process, then you have less intrinsic factor, and it doesn't help with B12 absorption. Now, I think one of the important things is to keep in mind here is I think there's typically about enough intrinsic factor in the gut, like during one meal, to absorb approximately 1 to 2 micrograms of B12. So there's not really copious amounts of intrinsic factor at any time. So it's important that the intrinsic factor levels are maintained at the highest level so that that B12 has intrinsic factor to interact with. The other thing is pernicious anemia is due to an autoimmune process, and it's age-related. So important point here to remember is pernicious anemia is an autoimmune process. In general, it's when our immune system makes antibodies that are targeted to different parts of parietal cells. So in other words, our immune system is attacking parts of parietal cells. And sometimes those antibodies are targeting the intrinsic factor itself or parts of the cell that make hydrochloric acid, and so hydrochloric acid production is decreased. But that also, there tends to be an increased risk that that's going to happen as we get older. Okay, so there are some other things that can cause B12 deficiency, like atrophic gastritis, which is inflammation of the lining of the stomach where it actually becomes inflamed and its function is compromised. But that wouldn't be pernicious anemia. That's a B12 deficiency. If it's an autoimmune process, then it can be considered that affects intrinsic factor, then it's pernicious anemia. Prevalence and treatment of deficiency. The incidence in the older elderly may be as high as 15%. Could even be higher than that. The B12 content of many vitamin supplements may not be high enough to raise plasma B12 concentrations. So oral B12 at least 6 to 9 micrograms, possibly up to 300 micrograms, appears to be necessary to correct the problem. So as we get older again, it becomes more difficult. Pretty good idea to take B12 supplements as we get older, but then also got to take high enough amounts. There's no UL for B12, so sometimes you're recommending a few hundred micrograms of B12 to try to overcome that deficiency of absorption of B12 that could be due to decreasing amounts of intrinsic factor or hydrochloric acid. Now, if that doesn't work, treatment of pernicious anemia or deficiency secondary to malabsorption often requires monthly intramuscular injection of the vitamin in amounts of 500 to 1,000 micrograms or oral doses of pharma dosages, which 2 milligrams, for example, the vitamin. So some people can still, if you take high amounts of B12, still get enough of it to enter into the bloodstream through the gut, but sometimes you have to go intramuscular injections where you're bypassing that lack of intrinsic factor or hydrochloric acid, and you're going directly into the tissue so the B12 is absorbed into the tissues and is functional. Okay. There's also, I think, some nasal gels where you can actually put it on the inside of your nose and the B12 will actually go through that epithelial layer into the blood. I think that's relatively effective as well. But we've probably all heard, people will say, oh, I get low on energy, and sometimes they'll go to a health practitioner and they'll say, oh, probably need a B12 injection. Let's give you one of those. They'll charge them a small amount of money, give them a B12 injection, and then sometimes the people will say, oh yeah, I always feel better after I get my B12 injections. Well, who would feel better? Maybe if you're low in B12, but until you actually have evidence to show that that's the case, probably not a good idea to just kind of get random injections of B12. Even though it's probably not harmful, you never know, but there is not a UL, so no toxicity, none's reported for that. So again, how do we assess B12? Well, if the classic symptoms are not present, then we're typically looking at serum or plasma B12 levels and measuring that directly, or sometimes methylmalonic acid is actually measured. All right, I think that wraps up B12. So I'm going to stop it right there.